re-emphasize these steps involved here again. So we want to think about the action potential being the first step, the action potential arriving at the exon terminal, which is step 1. Step 2 is the opening of the voltage-gated calcium channels in response to that action potential. Remember, these are voltage sensitive. So when the membrane potential triggers threshold, then we open up these channels that allows calcium to rush in. Calcium depolarizes the axon terminal membrane, which triggers the vesicles docking at the membrane and then dumping the neurotransmitter out into the cleft. Step 4 is that the neurotransmitter crosses the cleft, binds to its receptor, and that triggers a cellular response. And again, we're going to talk about the different types of cellular response based upon the different types of receptors. All right. After that, the response has to be terminated. Again, the signal has to be terminated by the removal of the excess neurotransmitter from the cleft. So there are basically 3 ways to remove that excess neurotransmitter. We can degrade it by the enzyme here on the postsynaptic side. We can reuptake and recycle it by the reuptake molecule on the presynaptic side. And then the third option is for it to be simply diffused away from the junction. So after it's broken down or in its whole form, it can just diffuse away, which is the third option for clearing the junction. Okay. Some of the differences here would be the synaptic delay. We talked about how the electrical impulse for a synapse is faster. The chemical synapse is a bit slower. And there's a delay about 0.5 to 5 milliseconds between when the action potential arrives and an actual postsynaptic cellular response. So the delay that we see between step 1 here and step 5 is really caused by the time that it takes for the vesicles to dock onto the presynaptic membrane and release that neurotransmitter. Okay, what the delay is not caused by is the time that it takes to diffuse. So diffusion happens pretty rapidly, but the delay is really caused by the docking of the vesicles and the exocytosis of the neurotransmitter. Okay, so really important to think about why this takes longer than even an electrical impulse is because of this delay where we need to dock these vesicles, then dump the neurotransmitter, and that takes time. Let's move on to our third lecture objective to compare fast and slow responses at the synapse. So going back to that fast and slow as the examples that I gave, bringing about the cellular response on the postsynaptic cell. Now we can think about channels acting as receptors, and that is going to bring about a fast response. So we're going to begin by thinking about what, excuse me, apologize. We're going to begin by thinking about what a fast response looks like. So a fast response is where the receptor is also a channel. We call this a channel-linked receptor. And this is called an ionotropic receptor. So basically, these ligand-gated channels are also acting as receptors. And remember, we talked about different types of channels. Ligand-gated means that something needs to bind to this receptor/channel, and that will open it up, and that will allow for a cellular response. So the channel is going to close as soon as the neurotransmitter leaves from that binding site. Okay, so when the neurotransmitter is not bound, the channel is closed. When the neurotransmitter binds, the channel opens up, ions begin to move, namely sodium and potassium, and that brings about a membrane potential change, and that brings about a cellular response. Okay, notice how this is faster because the channel is also the receptor. So the receptor, as long as it binds that ligand, it opens up a channel, and then we can automatically see that cellular response. We can compare that to a slow response. This happens if we have a metabotropic receptor instead. So a metabotropic receptor is called a G protein coupled receptor. This is a much more complex process. There are many more steps involved in the process in order to go from the binding of the neurotransmitter to the cellular response. So as we can see, this is a slower process than the faster ionotropic receptors. So if we think about the two different examples here, we have the second messenger system and then we have direct coupling. So let's look at how those differ. So with a second messenger system, the neurotransmitter binds to the metabotropic receptor. As we just described, the metabotropic receptor is coupled to a G protein. So here's the G protein, and the G protein has 3 different subunits associated with it. So when that neurotransmitter binds to the receptor, automatically the alpha subunit of this G protein is going to dissociate off and move downstream in the membrane and actually activate another enzyme that is in the membrane. This is usually referred to as an amplifier enzyme because it is going to amplify the signal and help to initiate the cellular response. So the alpha subunit activates the enzyme, and then the enzyme is going to generate a second messenger. That second messenger is what is going to help bring about the cellular response by opening or closing ion channels. And depending on the type of channel that we open or close, that will dictate if it is a depolarizing event that increases the membrane potential, or if it is a hyperpolarizing event that decreases the membrane potential. So the exact type of channel here will dictate if the cellular response is a positive change to the membrane potential, or if the cellular response is a negative change to the membrane potential. Okay. I hope everyone's with me so far. Please let me know if you have questions as we go through this, right? I know this can get a little bit complex. Okay. The second type of example here is direct coupling. And in direct coupling, the first few steps are pretty much the same. Neurotransmitter binds to our metabotropic receptor that is coupled to a G protein. The alpha subunit of that G protein moves off. And instead of activating an amplifier enzyme, it directly activates a channel in the membrane. So we sort of cut out the second messenger in this second example. And based upon whether this ion channel, depending on what type of ion channel this is, we can see a different type of cellular response, either positive or negative, depending on the channel. Also depending on whether we close or open the channel, okay? And that I'll talk about what that means coming up. So we can send a signal to open the channel or close the channel, and depending on that, as well as the type of channel, will dictate whether it's a positive change or negative change of the membrane potential. Okay. So let's talk about how we trigger graded potentials. So this is kind of bringing us full circle from when we began talking about graded potentials. So when the ligand gated ion channel opens up, the membrane potential will change. And remember, opening up of a sodium or calcium channel allows calcium to rush into the cell. So that is going to result in a depolarization event. And we can refer to that as an excitatory postsynaptic potential, which means that this change or cellular response that we're going to see on the postsynaptic cell is an excitatory change, right? This is also called an EPSP. Now, if we open up a potassium channel, we know that potassium likes to move out, or a chloride channel. Chloride is a negatively charged ion, so it's going to make the membrane potential more negative. So opening up of either of these channels will result in a hyperpolarization event, and that brings about an inhibitory postsynaptic potential, or an IPSP. Okay, so this is what I mean by the type of channel, whether it's sodium or calcium, based upon the ECDF of that ion, as well as whether we open or close it. We can think about these two principles in the reverse. So let's say I closed a sodium or a calcium channel. What do you think that would do to the membrane potential? Would it be more positive depolarization or more negative hyperpolarization. If I closed a sodium or a calcium channel, how do you predict that would change the membrane potential? Okay, so this would actually hyperpolarize the membrane potential. So I say that to say that we can think about these two principles in the reverse, that opening sodium or calcium depolarizes, but closing sodium or calcium hyperpolarizes. Similarly, for potassium and chloride, opening potassium or chloride hyperpolarizes, closing potassium or chloride depolarizes, right? Okay. Now, when we think about the postsynaptic potentials, this is going to be the change in the membrane potential in response to the type of receptor and the action happening when the receptor is bound by the neurotransmitter. So when we think about the excitatory postsynaptic potentials, the most common neurotransmitter is glutamate, okay? So glutamate binding to its receptor slash channel or its metabotropic receptor typically brings about an excitatory postsynaptic potential. Similarly, for inhibitory postsynaptic potentials, the most common neurotransmitter is GABA. So when GABA binds to our receptor, it's usually going to bring about an inhibition or a hyperpolarization, which is an inhibitory postsynaptic potential. All right. Okay. And so just to kind of explain the examples here, this means that when glutamate binds, the types of activities that we could see is opening up of sodium or chloride channels, which brings about EPSPs, or closing of potassium and chloride channels, which also brings about EPSPs. Similarly, for GABA, when GABA binds, the types of events or responses we would see are hyperpolarization. So that could be either closing sodium and potassium channels, sodium and calcium channels, or opening potassium and chloride to bring about that IPSP. So just to give examples of how these EPSPs and IPSPs are brought about. So like we mentioned, excitatory is a positive action membrane potential change, more likely to result in an action potential because again, we're raising that membrane potential, getting it closer to threshold, and that is a depolarization event. Inhibitory is moving us further away from the likelihood of getting to an action potential, which is a hyperpolarization change, a negative change in the membrane potential. Remember, if we move further away from that negative 55 in terms of being more negative, then that is making it harder to fire an action potential. Another type of event that we could see with inhibitory postsynaptic potentials is basically just stabilizing the membrane potential. So let's say we've had a lot of excitatory impulses coming in, an inhibitory impulse not only makes it harder to fire an action potential, but it could just cancel out the positive changes and just stabilize the membrane potential, making it or bringing it back to rest, right? Canceling out some of the positive changes. So let's look at some of the examples here just between fast and slow. So really just giving the steps that are involved in each. Let's start with the, fast response here. So the fast response, the neurotransmitter binds to the receptor. The receptor is also a channel, right? One and the same. That allows ions to move as the channel opens up, and that automatically brings about a response. The response will be determined by what ions are moving and whether or not the channel is going to allow the ion in or out, okay? However, the fast receptor is usually this example, which means that it's usually allowing sodium in, potassium out, which brings about a depolarizing event. Okay, so the ionotropic receptors and the fast receptors are usually going to result in depolarization as they automatically open up a channel, which means more sodium comes in than potassium goes out. Just thinking back to the ECDF here, remember sodium is a rush, potassium is a leak. So this gives an overall depolarization as more sodium rushes in. Okay. However, on the slow response, now is where we get into different types of responses depending on the exact channel and the second messengers. So just to go over the steps here again, the neurotransmitter binds, the alpha subunit moves off, the amplifier enzyme is activated, the amplifier enzyme produces second messenger, which is usually cyclic AMP, but it can be other things. And that usually results in a cellular response by way of activating a channel. And depending on what that channel is, and depending on whether it's open or closed, will determine whether an IPSP is created or an EPSP is created. Okay, so I say all of that to say a fast response usually automatically depolarizes. A slow response will really be determined by the exact channel and whether or not it's opened or closed. So thinking about the inhibitory synapses, we said that they can either be negative changes, which hyperpolarize, or they can simply stabilize the membrane. So if it's a channel for either potassium or chloride, potassium we know moves out according to its ECDF. So that is going to be an IPSP as positive charge leaves the cell. Chloride, on the other end, usually moves in, but it's negatively charged. So that is also going to result in an IPSP. Chloride can also have a stabilizing effect on the membrane potential. So we talked about this being a hyperpolarizing, but also a stabilizing because it can cancel out the positive charge inside the cell as well. So here's an example of a cell where there's no active transport of chloride, and we can see that there's equal positive and negative charge leaving the cell, right? So that is basically allowing for a depolarization. And then on a cell where there's no active transport of chloride, but now we're exciting the cell. So we have that same cell, we're exciting it, and now we're seeing that sodium is rushing in. You can see that positive charge entering the cell, but because chloride is also moving in, that simply cancels out that positive charge. And so we can see that there's no membrane potential change because it stabilizes that membrane potential by canceling out the positive charge. So we say that to say that chloride can help to stabilize or cancel out the membrane potential and keep the cell at rest.